This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas. I want you to close your eyes for a second and picture a scene from the deep, deep past.
Okay, setting the stage.
Yeah. So we're standing on the banks of the Angara River. This is a massive, incredibly powerful body of water flowing out of Lake Baikal in southeast Siberia.
Right.
The air is violently cold, just biting at your exposed skin.
Yeah.
And the taiga forest, dense stands of pine and larch and birch, it stretches out in every direction as far as the eye can see.
It's a gorgeous but completely unforgiving landscape.
Exactly. And the year is roughly 3,500 BCE, so we were looking at a world 5,500 years ago.
And, the landscape there dictates absolutely everything about human survival. The people living in the Angara River basin at this time, they're highly mobile hunter-gatherers.
Moving with the seasons.
Right, moving with that brutal rhythm of the seasons. They're pulling fish from the freezing waters of the Angara, tracking game through the taiga, just utilizing every single piece of the local flora and fauna.
And the really crucial thing to visualize here for you listening is what is not in this landscape.
Oh, yeah.
Because there are no farms. There are no sprawling, permanent cities walled off from the wilderness.
No infrastructure like we think of it.
Nothing. No domesticated livestock pens. Maybe just some working dogs. It is a life of open air, constant movement, and these deep, tightly knit kinship networks.
Yeah.
And here's where it changes. As we look closer at this specific moment in time, the natural rhythm of their lives is completely shattered.
The routine stops.
The hunting parties have stopped. The fishing nets are dry. Instead, families are frantically breaking the frozen Siberian earth. They are digging shared graves and, they are burying their children.
It's just a horrifying image. And for decades, archaeologists excavating these late Neolithic cemeteries around Lake Baikal, they could really only observe the quiet aftermath of this event.
Right, just looking at the bones.
Exactly. They could meticulously document the skeletal remains. They could note the really unusual clustering of young bodies and catalog the beautifully crafted grave goods left beside them. But the actual biological trigger...
Like what actually killed them. Right. The trigger of this sudden, devastating tragedy was entirely invisible to the naked eye. It just looked like a statistical anomaly in the dirt.
Well, we have a massive stack of source material for today's deep dive that completely unravels that anomaly.
Yes, we do.
We are diving into a really groundbreaking scientific paper published in the journal Nature, and the researchers here pulled off this incredible feat of microbial forensics.
It's basically ancient CSI.
It really is.
Yeah.
They managed to extract ancient, heavily degraded DNA from the dental cementum of these ancient Siberians. And buried inside that genomic data is a historical plot twist that completely forces us to rewrite the history of human disease.
It's massive.
So our mission today is to unpack the absolute earliest known lethal outbreaks of the world's most infamous pathogen, the plague.
Yersinia pestis.
Yeah.
The bacterium that would, eventually become the architect of the Black Death.
Yeah.
Finding it circulating in the specific population in this incredibly remote geographic region 5.5 millennia ago, it completely upends the traditional epidemiological models.
So let's talk about those models. Because finding the plague here shouldn't just be surprising. Based on conventional wisdom, it should be practically impossible, right?
Basically, yes.
Because think about the classic profile of a plague outbreak. We are completely conditioned to picture crowded, squalid, medieval metropolises.
London, Paris, Florence.
Exactly. We picture open sewers, grain silos, massive agricultural hubs crawling with rats, and crucially, as we'll get into shortly, crawling with fleas. These Siberian hunter-gatherers had absolutely none of those infrastructure elements.
None at all. And historically, the scientific consensus has heavily relied on what we call the epidemiological transition theory.
Okay, break that down for us.
Sure. So this framework posits that the emergence of severe epidemic-level zoonotic diseases jumping from animals to humans, is directly linked to the Neolithic agricultural transition.
When we started farming.
Right. The core assumption was that humanity had to fundamentally alter its relationship with the environment. We had to stop our nomadic movement, settle into high-density permanent villages, and accumulate massive amounts of organic waste before a pathogen like Yersinia pestis could sustain a catastrophic outbreak.
And, I mean, the logic makes intuitive sense. You start farming, you store grain. Grain attracts rodents. Rodents bring fleas.
Exactly. It's a very clear pipeline.
You live in a mud brick house with 50 other people and a dozen goats, and suddenly you have the perfect biome for disease amplification. But hunter-gatherers were thought to be largely insulated from this.
Yes, simply because they didn't sit still long enough to build up that critical mass of waste and pests. The prevailing thought was that even if early humans occasionally encountered what we call basal strains of Yersinia pestis,
Basal meaning?
in strains situated near the very root of the bacterium's evolutionary tree, sort of the earliest versions.
Right, okay.
Researchers assumed that the resulting infections from these basal strains would have been relatively mild. Without the dense urban infrastructure to support rapid transmission, and without specific genetic adaptations that defined the later medieval strains, they thought these early encounters might have just manifested as localized, benign, foodborne illnesses.
Like a bad stomach bug.
Yeah, perhaps an isolated case of severe gastrointestinal distress from consuming contaminated meat, but certainly not a society-collapsing pandemic.
But the data from this Nature paper completely demolishes that assumption.
Utterly destroys it.
The research team turned their sequencing tech toward 4 late Neolithic cemeteries in the Baikal region. That's Ostida, Shumilika, Bratsky Kamen, and Surovo.
Yes.
They ran shotgun sequencing on 46 individuals. And out of those 46 hunter-gatherers, they detected the plague in 18 of them. That is a 39% detection rate across the sample pool. That is wild.
To really appreciate the magnitude of that 39% figure, we have to look at the mechanics of ancient DNA recovery, because DNA is not a stable molecule over geological time.
It falls apart.
It does. The moment an organism dies, its DNA begins to fragment, driven by cellular nucleuses and just environmental water. Over 5,500 years, the human and bacterial DNA in those remains degrades into incredibly short segments.
Right.
Furthermore, 99% of the genetic material in an ancient tooth is modern environmental contamination.
Like dirt.
Dirt, soil bacteria, fungi, maybe even the DNA of the archaeologists themselves who dug it up.
Oh, wow. So they aren't just looking through a microscope and seeing a perfectly preserved, intact bacterium in there.
Not at all. They are sifting through millions of tiny, degraded genetic puzzle pieces and trying to reconstruct the Yersinia pestis genome computationally.
And that's the shotgun sequencing.
That is the essence of shotgun sequencing. They extract the DNA from the dental cementum, which is the calcified tissue on the roots of the teeth.
Why the roots?
Because cementum has this incredibly dense hydroxyapatite matrix. It basically locks in the blood-borne pathogens that circulated through the tooth's pulp during the victim's final days.
Oh, like a time capsule.
Exactly. It shields them from the worst of the soil's microbial activity. But even with that protection, finding a 39% positive detection rate is just staggering.
Yeah.
And it does not mean that only 39% of the population contracted the disease. It means 39% of the recovered remains were exceptionally well preserved enough to yield statistically unassailable genomic proof of the infection.
Okay, but hold on. I need to jump in on that 39% figure on behalf of anyone listening right now.
Sure.
Because 18 out of 46 sounds terrifying in a vacuum. But we're talking about remains pulled from 4 different cemeteries representing an entire archaeological period.
The late Neolithic, yes.
Right. So couldn't that 39% just represent a slow, steady trickle of endemic disease? Like if I pull 46 bodies from a modern city spanning 200 years and find that a percentage of them died of influenza?
That doesn't mean a single catastrophic flu pandemic wiped them out simultaneously.
Exactly. How do we know this isn't just a background illness they lived with over centuries?
It's a great question, and the research team anticipated that exact skepticism. To prove this was an acute epidemic rather than a slow endemic drip, they layered the genomic sequencing with incredibly precise radiocarbon dating and identity by descent kinship algorithms.
Okay, let's break those down.
Let's look at the Yersinia cemetery first, because that yielded the highest concentration of positive cases. They didn't just take the raw carbon-14 dates at face value. They utilized Bayesian chronological modeling.
Which means?
It's a statistical technique that takes the raw radiocarbon dates and constrains them using archaeological priors. Things like the stratigraphy of the graves, who is buried above or below who, and the distinct cultural artifacts buried with the bodies.
So it's narrowing the window of time.
Dramatically narrowing it. The Bayesian models demonstrated that the deaths of these plague-positive individuals at Yersinia didn't span centuries. Radiocarbon dates are exceptionally tightly clustered. They fall within a temporal span on the order of a single decade or two.
Wow, okay, so that eliminates the slow drip theory entirely. But decade is still 10 years. How do they prove they died at the exact same time rather than, say, a few years apart within that radiocarbon window?
That is where the identity by descent, or IBD analysis, comes into play.
Right, the kinship algorithms.
Exactly. IBD algorithms analyze the specific, unbroken blocks of DNA shared between individuals. Every generation, chromosomal recombination shuffles the genetic deck.
Right, from the parents.
Yeah. So by measuring the length and frequency of these shared unshuffled DNA segments, geneticists can precisely determine the degree of biological relatedness. They can differentiate between siblings, first cousins, or more distant relatives.
Because siblings will share massive, long blocks of identical DNA, while cousins will share much smaller fragments that have been broken up by an extra generation of recombination. Is that right?
That is absolutely correct. So when the team ran the IBD analysis on the Ida remains, they reconstructed intricate family pedigrees. They found sibling pairs, cousins, and crucially, an aunt buried with her nephew.
Wow. In the same grave.
Yes. Now, when you take that biological pedigree and overlay the osteological data, their estimated ages at the time of death, the statistical probability of a slow drip completely vanishes.
Right, because if an aunt and a nephew are buried together and both test positive.
The inferred age gaps at death perfectly align with a sudden, localized mortality event. We are looking at an acute, highly lethal strike that wiped out extended family units simultaneously.
Which introduces an incredibly dark mechanical mystery.
It does.
If these communities weren't living in dense cities,
Yeah.
and they didn't have massive grain stores attracting thousands of rats, how is a plague sweeping through a family unit in a matter of days?
Right?
Because the classic mechanism of the Black Death relies on a highly specific pipeline. The rat carries the bacteria, the flea bites the rat, the flea bites the human. If you remove the urban density and the rat infrastructure, how does the pathogen operate?
To solve that, the researchers had to conduct a deep bioinformatic analysis of the ancient Yersinia pestis genome they just recovered. They mapped the genetic architecture of this basal strain against the later, highly virulent medieval strains.
And what did they find?
They found a glaring absence. This 5,500-year-old Siberian strain lacked the IMDA gene.
Okay, let's focus on that missing IMIT gene, because in the source text, this seems to be the absolute linchpin of the whole discovery. What does that gene actually do in modern plague bacteria?
Well, IMTIX codes for Yersinia murine toxin, which is biologically a phospholipase D enzyme. In later strains of the plague, this gene is the absolute master key for flea-borne transmission.
How so?
When a flea ingests blood infected with medieval Y. pestis, the bacteria enter the flea's midgut. Now, the midgut is a highly hostile environment. It's full of toxic breakdown products from the host blood plasma.
So the bacteria should die.
They should. But the IMP gene produces an enzyme that protects the bacteria from these toxins. It allows it to survive, colonize the flea's digestive tract, and eventually form a massive biofilm plug in the flea's proventriculus.
A plug, like it blocks their stomach.
Exactly. It essentially starves the flea, sending it into an absolute biting frenzy. And that frenzy is what transmits the disease to new human hosts.
Man. So if a pathogen is a burglar trying to hack its way into the human ecosystem, the IMIT gene is the specific lockpick required to use the flea door.
That's a great analogy.
And because this ancient Siberian strain was missing the IMIT gene entirely, it was physically impossible for it to survive the flea's midgut.
Right.
So the entire bubonic transmission route, the flea bites, the swollen lymph nodes, the reliance on massive rat populations, all of that is completely off the table.
It is structurally impossible.
Yeah.
And for a long time, the absence of this gene in basal strains led epidemiologists to assume these early variants were ecologically trapped.
Because they couldn't hitch a ride.
Exactly. Without the flea vector, they couldn't spread efficiently, and therefore they thought they couldn't cause pandemics. But pathogens are incredibly resourceful evolutionary engines.
So it had a backup plan.
It did. This Siberian strain didn't need the flea vector because it possessed a different, profoundly destructive genetic weapon.
It had a completely different mechanism of attack.
Yes. While it lacked IMIT, its genome contained the UPM superantigen locus.
Okay.
This is a specific sequence of genetic material that Yersinia pestis inherited directly from its evolutionary ancestor, which was an environmentally robust gastrointestinal pathogen called Yersinia pseudotuberculosis.
Superantigen is one of those terms that just sounds incredibly dramatic.
It lives up to the name, unfortunately.
Break down the biochemistry for us. What does the EPM superantigen actually do to the human immune system when it gets inside the body?
Well, we have to look at how T-cell activation normally works. Under standard physiological conditions, your immune system acts with extreme precision.
Like a targeted response.
Right. An antigen-presenting cell will engulf a pathogen, chop it up, and display a tiny, highly specific peptide fragment on its surface using an MHC class II molecule.
Okay.
Then, a passing T-cell will use its T-cell receptor to inspect that peptide. It's a highly secure lock-and-key mechanism. If the receptor matches the peptide perfectly, that specific T-cell activates to fight the infection.
So it's very controlled.
Very controlled. Usually this process activates maybe 1 in 10,000 T-cells. It's a targeted sniper strike.
It's like a security guard meticulously checking a specific ID badge before allowing someone access to a building.
Exactly. But the EPM superantigen completely circumvents that security checkpoint. The superantigen protein does not go into the peptide binding groove at all.
Where does it go?
Instead, it binds to the outside of the MHC class II molecule on the outside of the T-cell receptor, and it mechanically clamps them together. It forces the connection regardless of what peptide is being presented.
Wait, so instead of a targeted security check, the superantigen basically acts like a rogue manager, chaining the door shut and tripping every fire alarm in the building simultaneously, just sending the entire security team into a blind, unstructured frenzy.
A catastrophic frenzy. By forcing these massive, uncoordinated cellular bridges, the EPM superantigen indiscriminately activates up to 20% of the body's entire T-cell population all at once.
20%? That's a massive difference from 1 in 10,000.
It is, and this massive cellular activation triggers the systemic release of pro-inflammatory cytokines, specifically interleukin-2 and tumor necrosis factor alpha. The immune system basically goes into a state of hyperdrive, resulting in a lethal cytokine storm.
Which means the body is attacking itself.
Yes. The inflammation becomes so severe that the body literally begins destroying its own vascular and organ tissues.
Wow, and the text makes a deeply disturbing connection regarding who is most vulnerable to this specific type of immune overdrive.
It does.
Because we know from modern medical observation that infections linked to this EPM toxin cause severe inflammatory complications, like Far East scarlet-like fever, which is also known as Izumi fever, and Kawasaki-like syndrome.
Yes.
And the demographic most heavily impacted by these syndromes today are prepubescent children.
The clinical data is unequivocal on that. Far East scarlet-like fever primarily targets children under the age of 14. And Kawasaki disease overwhelmingly affects infants and children under 5 years old.
Why is that?
Because a child's immune system is highly reactive and heavily populated with naive T cells, which makes it uniquely susceptible to the indiscriminate, overwhelming activation triggered by a superantigen.
Which means this 5,500-year-old pathogen was carrying an inherited genetic payload naturally calibrated to obliterate the youngest, most physiologically vulnerable members of these Siberian communities.
That's awful. And that molecular reality, the destructive capacity of the EPM superantigen is perfectly, tragically mirrored in the physical archaeological evidence at Lake Baikal.
Let's talk about the physical sites, because this is where the genomic data transforms back into a visceral human narrative.
Right, stepping away from the microbes and looking at the people.
Exactly. The researchers mapped the mortality profiles of these late Neolithic cemeteries against standard baseline burial data for the Baikal region. And the two sites with the highest concentration of positive cases, Ustaida I and Bratsky Common, stand out as extreme statistical outliers.
They do. In a typical prehistoric demographic model, you expect to see a U-shaped mortality curve.
Meaning a lot of deaths at both extremes of age.
Exactly. You anticipate high infant mortality, which was unfortunately a universal reality in the ancient world, and you expect a high concentration of deaths among the elderly population.
Right.
But the plague-affected cemeteries completely break that model. The data shows a massive, highly unnatural spike in the deaths of children between the ages of 7.5 and 11 years old.
And the researchers point out something heartbreaking in the pedigree groups. The parents are conspicuously absent from the simultaneous mortality events.
Yes, they are.
You have siblings dying together, cousins dying together, but very rarely parent-offspring pairs. The lethality was intensely concentrated on the children, which perfectly tracks with that pediatric vulnerability to the EPM superantigen we just discussed.
And the specific burials offer a really grim window into the speed and ferocity of the outbreak. At the Bratsky Common Cemetery, archaeologists excavated a shared grave containing 3 young girls.
3?
Yes. Their osteological profiles placed them roughly between 4 and 9 years old.
And the IBD genetic analysis, the kinship algorithms, showed they shared rare mitochondrial mutations, indicating they were almost certainly close maternal relatives, sisters or first cousins.
And all three of those children tested positive for the identical strain of Yersinia pestis.
Meaning they contracted it simultaneously, they died simultaneously, and the surviving members of the community had to dig a single grave into the Siberian permafrost to bury three little girls all at once.
It's hard to even fathom.
At the Osalita ice site, there's another double burial holding a nephew and his aunt, both of them positive.
So this pattern of synchronous death among close kin who were cohabitating, it solves the secondary mechanical mystery of the outbreak, which is the transmission vector.
Right, how it got from person to person.
Exactly. We established that the imp gene was absent, ruling out flea-borne transmission entirely. Without fleas, and given the rapid, localized spread among family members, the epidemiological evidence overwhelmingly points to direct human-to-human transmission.
They were catching it from the air they breathed.
It is highly probable that this pathogen manifested as pneumonic plague. When Yersinia pestis colonizes the lung tissue, it replicates furiously.
Okay.
The infection triggers intense coughing fits, and every single cough aerosolizes millions of bacterial cells into microscopic respiratory droplets.
So think about the environmental reality of a Siberian winter for a second.
It's brutal.
The community is huddled together inside heavily insulated winter shelters or tents just to survive the extreme negative temperatures. If one person develops pneumonic plague in that unventilated enclosed space, the air becomes instantly saturated with infectious droplets. The transmission to anyone else inside that structure, especially the children, would be immediate and utterly devastating.
Airborne transmission represents the absolute fastest, most lethal pathway for the plague. The incubation period is incredibly short,
Like, how short?
a matter of days. And without modern antibiotics, pneumonic plague is essentially 100% fatal within a few days of the onset of symptoms.
But, the source material highlights a detail regarding the aftermath of these deaths that I find incredibly profound.
The burials themselves.
Yeah. Despite the sheer terror of an invisible airborne killer tearing through the children of the community, there was no total societal collapse. We know this specifically because of how the dead were handled.
The mortuary data provides crucial insight into their cultural resilience.
Right, because if an outbreak is so catastrophic that it wipes out the entire adult population, the bodies are just left where they fall. There's no one left to dig the graves. But that didn't happen here.
No, it didn't.
The survivors didn't just scatter into the taiga in a blind panic. They stayed. And they meticulously buried their dead according to the deeply entrenched traditions of the Isakovo and Surovo cultural periods.
And we have to remember, digging a grave in the Baikal region isn't a trivial task. It requires immense physical labor to break through the rocky, frozen soil.
And they didn't just dig bits. They adhered to highly specific ritual orientations and included sophisticated grave goods.
Tell us about those.
Well, at Ustaita I, which aligns with Isakovo traditions, the bodies were carefully positioned parallel to the river. The survivors placed beautifully crafted, miter-shaped clay vessels with pointed bottoms into the graves.
Those vessels were designed to sit perfectly in the ashes of a hearth, right?
Exactly. And they buried them alongside intricately carved bone arrowheads. At Bratski Kamen, representing the slightly later Surovo influence, the bodies were oriented perpendicular to the river and accompanied by distinct egg-shaped pottery.
Taking the time to craft those specific vessels, to align the bodies with the flow of the Angara, while an airborne pathogen is actively threatening the entire camp.
Yeah.
It speaks to an immense level of social cohesion and devotion. They honored their dead, even in the face of absolute epidemiological terror.
It demonstrates that complex, resilient social structures existed long before the advent of urban agricultural centers. So we have a clear picture of how the pathogen operated.
The GPM superantigen triggering the cytokine storm in the kids.
Right. And we know how it spread airborne droplets in enclosed winter shelters. But the ultimate epidemiological question remains, what was the spark?
The origin point.
Exactly. If there were no urban rats bringing the infection into the camp and no fleas to act as a bridge, where did the first human catch it? What was the zoonotic reservoir sitting out in the taiga?
To find the reservoir, we just have to look at the local ecology of the Baikal region, which actually remains remarkably consistent even today. The primary endemic host for Yersinia pestis in this part of the world is the Siberian marmot, Marmota sibirica.
A marmot. So basically a massive, highly social ground squirrel.
Exactly. Marmots live in extensive colonies, and they are a vital ecological component of the steppe and taiga ecotone.
But how did the humans interact with them?
Well, for these late Neolithic people, marmots were an indispensable resource. The hunter-gatherers heavily exploited marmot populations. They hunted them for their calorie-dense meat, and they relied heavily on marmot pelts.
For warmth, I imagine.
Right, marmot fur provides incredible insulation against the brutal Siberian winters. The archaeological record from this region actually shows marmot incisors frequently used as personal ornaments and grave goods in earlier periods, which really underscores their importance to the local economy.
So walk us through the mechanics of the spillover event. How does the plague jump from a hibernating ground squirrel into a human lung to start a localized pandemic?
The spillover occurs during the intersection of hunting and processing. When a hunter traps or kills an infected marmot, they are immediately handling a biological hazard. There are two primary avenues for the pathogen to breach the human system at this point.
Okay, what's the first?
The first is gastrointestinal. If the hunter or the community consumes raw or insufficiently cooked marmot meat or organs, the bacteria enters the digestive tract, resulting in septicemic plague,
Septicemic,
a massive, overwhelming infection in the bloodstream.
which is going to kill the hunter, surely, but it doesn't necessarily trigger the coughing fits needed to start an airborne epidemic in the tent.
Precisely. It's a dead end for the bacteria, epidemically speaking. The second avenue is much more insidious and is the most likely catalyst for the human-to-human pneumonic outbreaks we see in the data.
The butchering process.
Yes. The skinning and butchering process is highly physical. As the hunter cuts into the infected marmot carcass, they are exposing highly vascularized tissue and infected blood. The mechanical action of butchering aerosolizes microscopic droplets of that infected blood.
So the hunter is leaning over the carcass, actively breathing in aerosolized marmot blood.
Correct. And the Yersinia pestis bacteria bypasses the digestive tract entirely and goes straight into the respiratory system. The bacteria colonizes the lung tissue directly. The hunter develops primary pneumonic plague. They return to the winter shelter, the pulmonary replication accelerates, the coughing begins, and the spillover event detonates into a familial outbreak.
What's deeply unnerving about this is that the data proves this wasn't a freak singular accident.
Not at all.
The researchers didn't just find evidence of one localized outbreak. By charting the radiocarbon dates across the 4 cemeteries, they identified 2 completely distinct phases of plague outbreaks. And those phases were separated by an astonishing 4 to 6 centuries.
That chronological gap is perhaps the most significant ecological finding in the entire paper.
Really? Why?
Because the fact that the pathogen emerged, caused a severe local mortality event, seemingly vanished from the human population, and then re-emerged half a millennium later with a genetically linked strain, it tells us everything about the bacteria's survival strategy.
It wasn't slowly circulating in the human population for 500 years.
No, the human population was too small and too sparse to sustain a continuous chain of transmission for 5 centuries. The bacteria didn't need humans.
It was out in the wild.
That gap proves the existence of a massive, robust, continent-spanning rodent reservoir completely independent of human activity.
The marmots.
Yes. The pathogen was thriving in the marmot colonies, successfully navigating the complex dynamics of marmot hibernation and local fleet transmission within the burrows, mutating and adapting in the wild. It was simply waiting in the ecosystem, causing localized, highly lethal spillover events whenever human hunters unknowingly interacted with an infected colony centuries apart.
It's a terrifying biological reality. You can live in total equilibrium with your environment, mastering the taiga, building a rich culture. And then one afternoon, a hunter skins the wrong marmot, and an entire generation of children is wiped out by an invisible airborne superantigen.
It forces a total paradigm shift in how we view epidemiological vulnerability. The narrative that mass casualty diseases are strictly the unnatural consequence of civilization, agriculture, and urban density. It's demonstrably false.
Yeah.
The Baikal data proves that zoonotic vulnerability is a universal human condition. Whether you are a Neolithic farmer densely packed into an early agricultural hub, or a highly mobile hunter-gatherer navigating the vast open expanses of the Siberian taiga, if your survival requires interaction with wildlife, you are inherently at risk of a spillover event.
Which brings this entire massive journey right back to your doorstep today, listening to this. Why does a 500-year-old genetic analysis of Siberian hunter-gatherers and marmots matter to a modern audience?
Right. What's the takeaway?
It matters because the fundamental biological mechanics of what happened on the banks of the Angara River have not changed one bit. The Nature paper explicitly highlights a staggering modern statistic. Today, 75% of all emerging human pathogens are zoonotic. They originate from animal transmission.
3/4 of all new threats. That statistic underscores that the most dangerous biological frontier on the planet isn't a lab. It is the physical interface between human populations and wild animal reservoirs.
We've covered an immense amount of ground in this deep dive. We started by dismantling the long-held assumption that catastrophic pandemics require the infrastructure of crowded, rat-infested cities.
We ventured into the fragmented DNA of ancient teeth.
Yeah, to uncover a forgotten microbial weapon, the EPM superantigen, that completely bypassed the need for fleas to launch a targeted airborne assault on the immune systems of prepubescent children.
And we trace the origin of that prehistoric nightmare back to the seemingly routine butchering of a Siberian marmot 5,500 years ago.
The synthesis of ancient genomics, rigorous radiocarbon modeling, and traditional archaeology gives us an unprecedented window into our deep past.
It illustrates that human history is inexorably bound to the microscopic world. And despite the sheer horror of these ancient outbreaks, the evidence of their careful burial practices serves as a profound testament to the endurance of human compassion and social resilience in the face of invisible threats.
It really is a remarkable testament to those communities. I want to leave you with a final, forward-looking thought to turn over in your mind as we close out this deep dive.
Oh, absolutely.
We have just examined how a small, sparse, environmentally adapted group of hunter-gatherers accidentally triggered a lethal multi-generational pandemic simply by utilizing the local wildlife for their survival. Consider what that implies for our reality right now.
Yeah, the modern context.
Modern anthropogenic climate change is rapidly and aggressively disrupting ecological niches on a planetary scale. Desperate animal populations are migrating to entirely new geographies to survive shifting temperatures.
And relentless human development is bulldozing deeper into previously untouched wilderness.
Exactly. We are currently forcing wild animals and dense human populations into closer, more frequent, and entirely unprecedented contact at a velocity never before seen in the history of the Earth.
The interfaces are colliding.
If the routine skinning of an isolated marmot could unleash this level of airborne devastation 5,500 years ago, what undiscovered, genetically novel pathogens are waiting out there in the shifting wild right now, quietly adapting in their hosts, just waiting for the perfect moment to cross over into their very first human lung?
Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.